departments of neurology at Massachusetts General Hospital and Harvard Medical School in Boston. She earned her master's in public health and PhD in biostatistics from Boston University, where she worked on predictive modeling of disease risk. And since she has published over 100 papers in multi-omics and genetic epidemiology of primarily neurodegenerative diseases, but she has experience across many different aspects of mental health. In addition to her research, Lori is internationally known for her training programs in biostatistics and her innovative teaching techniques. Specifically, Lori has taught me never to underestimate the incentive power of candy, and I've tried to use that in all of my teaching. And for example, while at Boston University, Lori managed a 6-week Summer Institute of Training in Biostats course, or SIBS, which was funded by the NIH. She's developed and taught multiple courses on research methods in biostats and statistical programming in sub-Saharan Africa. She's currently the principal investigator of the Global Initiative for Neuropsychiatric Genetics Education and Research, or GINGER, based out of the Harvard School of Public Health and the Stanley Center for Psychiatric Research at the Broad Institute of Harvard and MIT. I personally know that Lori is a champion of diversity and equity in everything she does, but notoriously she puts her money where her mouth is, and we're very fortunate to have Lori here with us today. Thanks for coming.
Thank you so much for that wonderful introduction, Dan. And thank you, everyone. I'm sharing my screen. Thank you for inviting me to give this talk. You said it's the first talk of 2020. I will say it is, I am very emotional and happy to say that it is the first talk of the Biden administration for your Southern neighbors. I warned Dan that the last hour and a half has been an incredibly emotional and moving experience down here in the US. And I think some of that emotion is still in me that is about to come out as I talk about one of the things that I'm really passionate about and I've done a lot of work in, and I would like to share with everybody. I titled this talk, Tackling Disparities in Genetics Research, Building Research and Researchers Together. And I've approached this in a way of looking at two different challenges. One is mental health disorders, and the other is global disparities. We talk a lot about disparity. The conversation has blossomed even more, and in a good way, down here and in academia throughout the world recently. In general, at least in my experience where I am, a lot of the diversity conversations are usually thinking in terms of a domestic audience. And I tend to think of diversity on a global scale. So think globally and act globally. I spoke with Dan yesterday, and he introduced me to the acronym that you guys have, which is EDI, of Equity, Diversity, and Inclusion. So I've kind of thought a little bit about, as I talk, how I hit on all three of those and how important keeping those three things in mind as you decide and work towards your research goals is. So I'm going to start, I think I'm preaching to the choir, so I'm going to kind of go through the impetus a little bit faster than I usually would, but I think this audience is very familiar with the fact that neuropsychiatric disease burden is the largest disease burden. These numbers are pre-COVID. So pre-COVID, the largest disease burden that we have been dealing with. These numbers still exist, and the COVID epidemic has just worsened a lot of the mental health disorders. But even though they take up about 28% of the years lived with disabilities, there's not as much research in it as you would think, certainly as compared to things like cardiovascular disease and cancer. And even so, a lot of the research that's been done, there hasn't really been a new mechanism of action in terms of drugs for severe mental illness. I'm talking here more about psychosis and schizophrenia, although there have been some new drugs that come out, just a whole different way of looking at them. We kind of stalled back in the 60s. And then for those who aren't interested in genetics, I usually have to point out that schizophrenia is actually a fairly highly heritable disease. On the x-axis, you have just prevalence, whereas on the y-axis, you have heritability. And we see up here in the upper left-hand corner bipolar disorder, schizophrenia, autism spectrum disorder. So many of these mental health disorders are highly heritable, so genetics can offer us some insight into that. Colleagues at the Stanley Center have really been looking at schizophrenia for a really long time and trying to use genetics to help understand it. Over the years, we've had multiple GWASs, and as those numbers of people that are included within the GWAS goes up, we get more and more cases. This was all done, work by the Psychiatric Genetics Consortium, bringing together as many cases and controls as possible. I believe this is from 2014. And, you know, 128 SNPs that were published back in 2014. We have more now. This is all great, and it's definitely leading us towards new discoveries and new insights. However, the thing I want to talk about is this. While this has led to wonderful discoveries, on the right, what you're going to see is the percent of the data included in that first big GWAS. 96% of the data came from people of European descent. On the left, what we have is a rough estimate of the breakdown of the global population. And as you can see, in the global population, only 16% identify or have some, identify as European ancestry. We're leaving out 84% of the population when we're doing all of this work. Now, they've improved since the first time I've ever given a talk like this. It's definitely improved. But, you know, the focus really needs to not just include diversity in all other aspects, but diversity within the actual data. So this is on bioarchives or medRxiv, sorry. And the most recent GWAS from the Schizophrenia Consortium. They've upped the number of cases. They've found now 294 SNPs that will offer some great insight, and they've definitely improved. It is now only 80% from European descent, but the other 20% from East Asian, still leaving out a vast majority of the world's population. And I don't mean to pick on schizophrenia, and I don't mean to pick on, you know, psychiatric data. This is true for most GWASs. I've seen this paper quoted so many times now, which I'm so happy that people are starting to really recognize, is that up until 2016, 81% of the data we were looking at came from people of European population. A shout out to my colleague, Alicia Martin, who has a much better sense of visual graphics than I'll ever have. She's put this together, and it's in a paper in Nature Genetics now, of really how much data that we use in GWAS coming from European population. So the top graph here just shows cumulative data. So in 2018, we still have quite a majority of our data coming from the European populations, even though here on the right, you can see the actual breakdown of European and Asian, South and East Asian, et cetera, populations. This is a fraction of each GWAS. And as you can see, there was a lot after that Popjoy paper came out. People were really trying to like up diversity in GWAS, but that's pretty stagnant lately, and it's sort of leveled off. And you know, this just isn't good. It isn't good for a lot of reasons, but what I would like to focus on is the scientific reasons why this isn't something we can continue. You know, we're clearly making discoveries. Why do we need to study diverse populations? Again, from what I understand in this crowd, there's a lot of knowledge about genetics, so I won't spend too much time. But in general, there's just more information in the African genome. The y-axis is number of variants per genome. So I usually translate that into audiences that are unfamiliar with genetics and genomics as amount of information within each genome coming from different populations. And as you can see, you know, the European populations are down here on the left. With my family happens to live in Finland, they tend to be the least amount of information, something I tend to tell them quite often. And then you have the African population over here with the most amount of information contained within their genome. Not only the most amount of information, but the most amount of rare variants as well. So here in this particular plot, we're actually looking at not just the diversity within the genome, but those rare variants that you can see within the genome. And again, you can see the African populations are the ones that have the most rare variants within their genomes. Discoveries that we make within using African genomes can turn around and help everybody, right? So there's more information. It doesn't mean that discoveries we make within diverse genomes only help those diverse populations, because, for example, one particular mutation that has led to a therapeutic drug to lower cholesterol would never have been found if the only thing we were looking at was European Americans. It's a particular mutation that's found in 2% of African Americans, but less than 0.1% of European. This drug now is being used quite prevalently in many populations. The other is fine mapping. This is work done by, I stole this slide from a colleague of mine, Hailiang Huang, who does wonderful talks on this sort of thing, such as fine mapping, is that Europeans have large haplotypes, East Asians have large haplotypes. In the end, what we want to do is we want to be able to find a causal variant. We might be able to say this section of the genome is associated with the disease, but we really want to be able to fine map it down to a causal variant. This is great. He likes to talk about it in terms of overlapping European and East Asian. I took this one step further and said, one of the other aspects of the African genome is that their haplotypes are smaller. And so if you add more and more diversity, it really does help us try to find and target in on that causal variant, which might give us more insight into function. And so that's my little 2-second talk on fine mapping. So there's a lot of reasons to want to include diversity on a scientific level. There's another reason, the Human Genome Project took 15 years and $2.7 billion. Now it costs about $100 to $1,000 to sequence a single genome. There's a lot more stuff that we can do. It's been a wonderful trip, and I feel like I've gone along on the ride with the human genome and seen from when we were just doing Sanger sequencing and genotyping and like little tiny things to now being able to map an entire human genome. And it's great. It's allowed us to elucidate functional pathways. It's allowed us to better predict things, to start talking about personalized medicine. The problem is, and that's the area that we're going in. This is a paper that came out a few years ago now, talking about personalized medicine and genetic risk scores being brought into the clinic to help us within the clinic. The issue is that it's not helping everybody. There's a huge segment of the population being left behind. So we are building these prediction models and these genetic risk models on this data that is primarily of European descent. So medicine is getting more precise for white people. A paper that came out in 2016 used an example of hypertrophic cardiomyopathy, where, at that time, they say, here's this mutation, or it's actually a couple of mutations, and if you have this mutation, you're going to get, or you're going to have an issue, that might end up with a heart attack, etc. Fair warning, I'm not a clinician, so forgive me to all the clinicians in the audience. And that's what they went on. Like, if you have this mutation, this is what you're going to have, because everybody in our data who has this mutation ends up having a hypertrophic cardiomyopathy. And the problem is they didn't have any African Americans in their data sets. So while those mutations that are listed on the screen here were almost predictive or deterministic for hypertrophic cardiomyopathy within a white population, they're actually fairly prevalent within, in this case, a Black American population. So they were misdiagnosing people and telling them, listen, you have this like heart condition and this is what you should do, etc., because of these mutations, when in fact they were actually fairly common within that population. And this caused a bit of a stir. I will say one of the things I love about this paper is just learning how things get classified. And after going through all of this, if you look at the bottom, they were saying 4 of the 5 variants remain classified as most pathogenic category of disease-causing mutation, and one variant was downgraded to disease-causing question mark, because apparently disease-causing and disease-causing question mark are two actually different designations. I did not know that. So, so to get back to the idea, that's a single mutation, or a single group of mutations, I should say. The idea of these genetic risk scores, which I think many people have heard about, polygenic risk scores and genetic risk scores, a lot of them are developed and they show a lot of promise, right? There's a lot of information in our genome. If we can come up with some sort of a score to say this person's at high risk, this person isn't, it would be a great asset. Again, this is work by my colleague Alicia Martin, who showed quite clearly, and she's got multiple papers on this, this is just one, that genetic risk scores developed in a European population work really well in a European population. If you apply that genetic risk score to, say, a South Asian population, an East Asian population, or here an African population, the prediction accuracy just goes down quite a bit because it was developed within a European population and applied to a different population. So if we keep pushing these genetic risk scores on the data that we currently have, the diversity, all we're going to do is end up leaving a huge population of the world behind, and we don't want to do that. And so again, I said there's many papers on this particular subject. These are just a few that I would share. So the question is who's currently doing this work? Like, I'm telling you, it needs to be done. There are people that are slowly starting to build up this sort of work. And about 8 years ago now, I think, they started H3 Africa, which is a combination of funds from the NIH and the Wellcome Trust. There's an African Society of Human Genetics, an African Genome Variation Project as well, to start funding more studies doing genetics in Africa. So, and there has been a lot of work also in other areas. I'm going to come back to our mental health, of our area of interest, which is mental health. But if people want to know about some of the other work going on in Africa, this is an excellent review paper that came out this year. So here's the issue, though. Only 4 of the 39 studies that were funded by H3 Africa in its latest round had anything to do with psychiatric disorder. Only 1 on schizophrenia. And the first round of grants that they put out, the first 5-year cycle, had no grants funded on any mental health. Eventually, they argued with them enough to get this particular project that I'll talk about for 2 seconds, called the Genomics of Schizophrenia in South African Xhosa people, funded, but mental health just doesn't make the top of the list, unfortunately. So that's one of the things that we've been pushing quite a bit. So this is called the Sac Study, and the paper came out this last year with many of our colleagues in South Africa, including some of my fellows as co-authors, which was quite lovely. So I'll talk briefly about what we're doing at the Stanley Center for Psychiatric Research. If people are unfamiliar, it's part of the Broad Institute. It was started by a generous gift from Ted and Vada Stanley, who have a son with bipolar disease, and the goal of understanding disease mechanisms to identify biomarkers, but really most importantly, identify new therapeutics. Under the leadership of Steve Hyman, the Stanley Global Collections was started in about 2012. It expands across many different countries. I am mostly involved with the Neurogap Project, which is South Africa, Kenya, Uganda, and Ethiopia, to expand the collections of genetic samples from these 4 populations. We're studying schizophrenia and bipolar. The USPI, and this is the US team, I'll show you the international team in a second, it's run by Kirsten Conan. We have a group of of PIs on the ground at our different sites, again, across these 4 particular countries. This collaboration was started through many, many years of getting to know people and building trust across different groups of people. The PI, Kirsten, had worked with Solomon Tefera before. Lukoye Etwale was one of her students. Lukoye then recruited Dickens Akena, who was one of his students and one of Dan Stein's students. So there was a lot of many years of work building up trust throughout this group in order to be able to put on this project. Very briefly, we are posed to collect 17,000 cases and 17,000 controls. It's a case control study. The consent is using something called the UBAC, which is another talk that I would give, but not at this moment. How do you consent people into a study for genetics when the word for genetics doesn't exist in the local language? The UBAC is, the research assistants talk the participant through what's going to happen in the study. Then they ask them 10 questions. And if they score high enough, showing that they understand what they were told, then they're consented into the study. We're collecting many psychometric measures and doing chart reviews as well. And we have a protocol paper here in BMJ open if you want to learn a little bit more about the study. DNA is collected in country. Sorry, samples are collected in country and DNA is extracted within the country. One aliquot of the DNA is then shipped to the Broad, where we are doing, we started by doing a global screening array, but we are switching to sequencing of all the samples. Each country also has all of their samples in a biorepository to be used for other things that they would like to use them for. One of the reasons we're actually shipping the samples to the Broad is so that we can do sequencing in bulk. We have more capacity at this point. That is, building up enough trust to be able to take the samples off the continent. Again, required a lot of working together and bringing together all the people, all the players within this particular study and to get that done. I can't remember if I got rid of the slide on that. I'll see in 1 second. We have set up actually labs to do the DNA extraction and train people to do the DNA extraction in each of our 5 sites. We have 2 sites in Kenya, one in Ethiopia, one in South Africa, one in Uganda, and then obviously collection sites throughout all the different countries. Here's the slide. It's something to be excited and proud of what we were able to put together, but it's also something that we need to be realistic about. To be able to put together a study that truly is a true collaboration with equity. It was 2.5 years to get the protocol approved and 12 IRBs because each country had its own IRB. And sometimes it wasn't just the institute, say Addis Ababa University, that we needed to have IRB approval from, but also the Ministry of Health in Ethiopia required IRB approval as well. So this isn't a small endeavor, and I'll be the first person to admit it, but it's absolutely worth the effort. And we have, I will say we are halfway through our collection, so we've had over half of the samples collected. It was stopped for a while during COVID, but the sites are slowly starting to get back up to collecting. But one of the reasons that Neurogap has been a success is that we've really built it with the idea of equity, diversity, and inclusion in mind. So how do you go about building an international collaboration that includes sites with differing resources? I'm going to say that a lot of the stuff that you'll see me talk about and a lot of the things that I learned just to give credit where credit is due as a Peace Corps volunteer. So that's a little bit of my background and also the requisite embarrassing picture. I was a Peace Corps volunteer more than 20 years ago now, which is a really hard thing to say out loud. I helped start a camp called Camp Glow, which was a girls' camp, which I'm happy to report is still going on today. That's me in the center and what I look like without hair product. Okay. So how do we go about doing this? We want to build a collaboration between institutes that have varying different needs and abilities to do. And so we need to always keep that in mind. And so this slide here always gets people, and it brings us back a little bit to the mental health aspect, is This here is the number of adult psychiatrists in varying countries across sub-Saharan Africa. So for example, in South Africa, there's about 207. In Uganda, it says 34. One of my Ugandan colleagues, when he was here giving a talk, used to love this slide because he'd say, well, I'm here, so it's only 33 now. Whereas in Massachusetts, the state that I call home right now, there's 831. So how do we work with researchers on the ground when there is such a disparity and such a need for psychiatrists to be in the clinic? So in Africa, on average, there's 1.4 psychiatrists per 100 million population, whereas in the US, it's 124. And I apologize, I should have looked up in Canada, I forgot. These numbers are getting bigger, but very, very slowly. So it's not just the psychiatrists that are lacking, it's also the researchers. So on the screen now is a slide. It was the first slide in the American Society of Human Genetics conference in 2016. And it was saying, look at this, isn't this wonderful? We have researchers here from all over the world. When this slide got put up, my phone exploded because every person that has ever known me sent me a picture of this slide saying, this is what they're saying, they have people from all around the world. And when I look at this picture, what I see is a gigantic part of the map that's missing so many people. So we're not just failing on who is included in our research, we're failing also on who is doing the actual studying. And this is something that I have spent a better part of my career trying to work and fix as best I possibly can. One of our fellows, and who is now the research director of one of our studies in Africa looking at autism, Celia Vandermeer, and I are working on a paper where she, instead of just looking at who's doing the research, we actually are working with Sanger looking at who is applying to use open access data. So who is sending in all those applications to be able to use data like UK Biobank and any of the open access data. And this is the most recent numbers that we had from about 6 months ago. And as you can see, there's no applications also coming from the continent of Africa. Also, this another paper for anybody interested in why we should be looking at African genomes. There's a paper from 2017, Gene Discovery and Complex Traits, Lessons from Africa. It's a really interesting paper. I mean, it's a great tutorial. It gets the point across, and for that reason, I like that aspect of it. But if you notice and you look at who the authors are on the paper that is called Lessons from Africa, not one of the authors are actually from Africa. And while I celebrate what's written in the paper, I actually have a very hard time with that at this point. These are authors that actually were co-PIs on this particular study that I talked about earlier, the Sac Study. They mentioned the Sac Study within that paper, but they did not engage any of the local researchers when they were actually writing the paper. And this is one of the problems. The model that we're used to is this colonial model of research, sometimes called safari research. You fly in, you harvest your data, you go have a good meal, and I'm sure Dan can tell you that good meals are abound on the continent. Go on safari, fly home, write your paper, never to be heard from again. And that's not a collaboration. We can't perpetuate that model of collaboration, and it has to change. The random African proverb of if you want to go fast, go alone. If you want to go far, go together. I sought to try to figure out what the origin of this actual proverb is. And I will say it takes a village to find the origin of a proverb, and no one can tell me. But I still think it's a great proverb, whoever came up with it. So with that in mind, I proposed and built GINGER, which stands for the Global Initiative for Neuropsychiatric Genetics Education and Research. Will always be referred to as GINGER because no one wants to say that whole thing, with the goal of building collaborations, training the next generation of research, and ultimately reducing the burden of serious mental illness. Building capacity by investing in people. Our 3 pillars are building support from above, building support from within, and making sure building support to the next generation as well. it was, you know, as I said, I learned a lot from my Peace Corps experience. And one of the things I learned is that you spend a year listening and then you spend a year working together. So, because Peace Corps is 2 years, sorry, should have said that. So I did. We spent 6, 8, 10 months just traveling the continent, meeting with our collaborators, meeting with the students, teaching the random class here or there. This is an R course we taught at Addis Ababa University. I believe this is in Uganda. I'm not sure. It looks like it's Uganda. We did some talking. I did a lot of listening, just meeting with people. What does it take to be a successful neuropsychiatric genetics researcher here? Who is doing this sort of research? How can we work together to build this particular community? We also hosted something called a curriculum Jamboree, where we brought everybody together to Boston in February. So I will say right now that shout out to all of our African colleagues that were willing to fly to Boston in February. 2 weeks after that travel ban. It was an interesting experience. It was a wonderful experience. And we spent 2 days just talking about what belongs in a curriculum, how to interact with students, what works for them, what can we provide, what can we do together. One of the things that came out of it were the top topic areas in each of our, what we think of as the main goals of what we wanted to do: epidemiology, biostats, genetics, neuropsych, and ethics. And from there, we held Ginger. Ginger has and had 3 aspects to it. It has workshops, it has virtual classrooms, and it has on-site training. And what you see here was Ginger, the first iteration. We are now in the second iteration. This is our first group of Ginger Fellows, 17 of them from diverse backgrounds. Some come from a neuropsych background, some are clinical psychiatrists or psychologists, one is a nurse, and a few of them are genetics. They were all recruited. They had to be nominated by one of our PIs. When I talked to our PIs about it, I said, you need to give me the people that will carry on your research. One day you will die, because we all know none of us will retire, and we want somebody to carry on our research. And we had applications, we reviewed them, and selected our 17 fellows. The Gingers by the number slides, our output over our first cycle was 17 fellows. They came from various backgrounds at that point. So some had not yet started a PhD, some were doing a postdoc, some were PhD students. So over the 3 years, we've had multiple, 3 people get started on a new PhD, 3 PhDs earned, promotions, fellowships, 70 papers, including multiple collaborative papers, 126 hours of virtual classrooms, 160 hours of workshops, 52 invited guests from all over the world, and then teaching fellows, which I'll talk about in a second. We put on 6 on-site trainings. When we do our on-site trainings, we bring in local TAs. Those TAs have gone on to reteach the material that we've been doing. So that's our first iteration. In the interest of time, I'm gonna just breeze through because everyone likes pictures, myself included. We do this in 3 different ways. We do workshops for our fellows, we do virtual classrooms, and we do on-site training. The workshop is our way to bring people together, so that's support from within, to build a community and to do slightly interesting, different types of education. One of the important things is bringing in speakers who our fellows can aspire to be. This was very important for us. As I looked back on my own career, who were the people that inspired me the most? Who were the people that I would look up to and say, that's what I want to do, that's what I want to be? And invariably, they tended to be white women. And, occasionally a guy would maybe slip in there as well. But they tended to be people that looked like me. And I don't think that has anything more to do with that. I could see this person and say, this is who I want to be, because I can see myself in them. So it was really important for us to bring in people that, you know, it's great to have people like Eric Lander come and speak and give these like phenomenal talks and whatever. We definitely did that. But I wanted them to also be introduced to people who came up the same way they did, that they could look at. And one of our speakers brought everybody to tears, which, by the way, was just so wonderful, talking about growing up in a village and how he got out, you know, in Ethiopia and how he got to where he is. It was wonderful. And then also we get to, like, do you remember back when we used to be able to hug each other, do things together? So we got to do that sort of stuff too, which of course is always fun. And, you know, the best way to bring people together into a scientific collaboration, if you ask me, is food. So there was a lot of food, there was a lot of celebrating. We had Ethiopian food, which is what we have here. We had Kenyan beer, we had South African wine. The Ugandan restaurant never returned our calls, so unfortunately we did not have matoke, but we brought together people within the Broad and Harvard with free food, free drinks, come talk, come meet, and come collaborate. And this is really how we brought everybody together, not just our fellows, but the postdocs and the researchers at the Broad as well. Here in the bottom left-hand corner is one of our fellows, Simon Kariuki from Kenya, and Dennis Lal, who is now at the Cleveland Clinic, and they've gone on to write grants together that have been successfully funded. Here we have a couple people that went on, Alicia and Andrea, who went on to be group leaders and help publish papers with some of our fellows as well. And then they got to tour the Genome Platform. We also put on one in London, which Dan wonderfully volunteered to come and help teach, which was great fun. And, you know, we talked about, we were able to do stuff like public speaking and negotiating and ethics workshops, and also get started on some of our group projects. And then, of course, we had fun. We played lawn games. We went to some football stadium. I don't remember which one because I don't like football. Arsenal, maybe? I don't know. I apologize to football fans in the room. It happened to also be the weekend that our former, oh, sounds so nice, former was in London, and so we all went and joined the protest, and that was fun. We also were able to get started on some of our group projects. We brought people in from the Broad to work as group leaders down on the bottom here, and work with our fellows to start group projects. Number 4 just made its way up on medRxiv, not bioRxiv, and a few others are in the process, are in the phase of getting finished up right now. We also did virtual classrooms, which I no longer have to explain what those are, so I'm going to skip it. Thank you. Everyone knows what a virtual classroom is now. And then lastly, we did on-site trainings as well, where this is support, where I said support from above, support from within, support to the next generation. We were able to work with people at our different sites to teach classes that they wanted, that they needed, room capacity. Our Ginger Fellows came and helped us. Our local TAs came and taught a little bit further. The teaching technique was always see it, do it, teach it. Anything more than 20 minutes, no one wants to listen to. I apologize, as I'm now on minute 40. We work together to do this at very many institutions, and this is where I get to show a picture of our teaching fellows, and you'll recognize at least one person on the slide. It gave me the opportunity to bring in postdocs, grad students, and junior faculty from our local here institutions to go help teach and to work with our fellows abroad, and has really built up a lot of friendships between. And I will say this is my chance to give a shout out to your own faculty member, Dan, who I am very possessive of my on-site classes. It is one of the things that I have worked on since the time I was a Peace Corps volunteer, not obviously biostats at that point, but it's my baby. It's so much of what I've done and I've built over the years, and I have hesitancy to let other people take it. And Dan was the first person I allowed to teach my class without me being there. And he showed up in Uganda a week before I did. And I showed up a week later and he had already given out way too much candy. He doesn't understand that the only reason that it's useful is if you don't do it every day. Anyway, and so I will say, and Dan had taught with me before in South Africa, which is why I knew and I saw how he was able to teach. And I was comfortable, albeit it was weird, allowing him to take my class over with me not there. And the students loved him. It was a great time for all. Just because I want to throw some pictures of all the different places that we teach. This is one in Kenya. And we did one in KwaZulu-Natal, a place where Dan has taught with me before. And this is a picture on Women in Science Day. It was quite lovely. Here's another picture with my signature candy, which are M&Ms. So we graduated in the virtual world, as many of us are used to, our 17 fellows, and it was Dan, as you can see on the picture, was there, with the same background. It was a very moving experience. I think it went pretty well, and I'm happy to say that we have been funded to do another cycle. So we have started our second cycle of Ginger. This was the original plan. It is funded partially by NIH and partially by the Broad Institute. We have 7 new fellows from our 4 different countries. COVID has shifted things a little bit. So we have not been able to put on our workshop yet in Boston. That is now scheduled for 2022. We are hoping to schedule a workshop somewhere in Africa in 2021. That remains to be seen. And in fact, I was supposed to fly to Uganda tomorrow. That's not happening for various reasons. But all of this will be put on, just shifted a little bit to the right. And in this time, our goal at the end is to have everybody be able to submit career grants. 2 of our fellows from the first cycle were able to do that, one of them successfully so far, and more on the way. I will say that we've focused a lot on our virtual classrooms in the last year, which I think is not surprising. Of all my colleagues at Harvard, I think I was one of the few that when we had to switch from in-person to online in a week back in March, I was the only one that didn't fret because I've gotten used to teaching online now. But we've learned a lot over the last few months on how to change up the virtual classroom and make them really useful. We do a 1 week on, 1 week off system as in that. I also, and I'm happy to talk more about that as well. We're also going to start putting these more online. So we're going to have content available just for our Ginger fellows, and then content available outside of Ginger, which I will send around to everybody that can have access to. These are topics like genetics, biostats, epi, but also neuropsych, and a few other things. Quickly, as I reach my time, I will say challenges and lessons learned. The heterogeneity of fellows was wonderful, but it is a challenge. It does make it a little more challenging. As I said, the second cycle of Ginger, we would like people to be able to submit a career grant at the end, which means the people that we recruit need to be at that point in their career. So having people who don't have their PhD yet with people who are writing career grants was difficult. And then everybody's coming from a slightly different environment. I also need to point out, because I don't think I say this enough, and I love our fellows, is that all of our fellows had and have full-time jobs while taking part in Ginger. None of them are full-time students. It's just not really heard of at that point. Many of them are balancing their clinical work with whatever else they have, and then on the weekends and on the nights taking part in Ginger. And it just boggles my mind. I'm always so thrilled to see how much energy they're able to put into it, and I love them for it. The bandwidth is very specific to actual bandwidth. That is always a challenge. One of the fixes we do is our videos have to be downloadable, not streamable. Not only streamable, they have to be able to download it because they don't have the bandwidth to stream it. And then challenges such as this last week, Uganda had a very contentious election and shut off the internet. Ethiopia is going through some issues right now, and they occasionally shut off the internet. So these are situations that we aren't used to dealing with, but we need to always keep those in mind. And I think pretty much lastly, I would actually say that before COVID, I used to have to argue for being able to do global research, that yes, you can Zoom with people and still maintain a connection. Yes, it's totally possible to be on 2 different time zones and be able to do it. And not so much have to argue for the in-person stuff. And that COVID has switched that so much. People now understand, oh my God, you can actually do stuff via Zoom. You can actually maintain connections that way. Although it's not that easy. That I feel like I'm now starting to make sure that I remind people the importance of face-to-face. So Ginger and Neurogap never would have happened if it wasn't for all the face-to-face that we had. And one of the things I've been feeling quite a bit with COVID is the lack of real connection with my fellows, because I would spend a lot of time in Africa, working with people, understanding. So I feel so distant at this point, understanding what they're going through, because I haven't been able to travel there. And hopefully with this, the new vaccines, that will come and that will happen. So I just want to thank Ginger. We have a website. Feel free to contact me with questions. We're a big group. This could not be done, certainly not without Christy and Courtney, as Dan will hopefully attest to, and Bazoo, who's our associate director and faculty. And acknowledgements, again, this is a large collaboration with people all over the world. And I like to end a lot of my talks with a quote from Lakoya Twale, who is now the dean at Aga Khan University, formerly at Moi University. This was something he said at our curriculum jamboree. And I think it really sums up the whole equity, inclusion, diversity notion that we've been talking about, which is our partnership is built on the principle of equity, to each according to their need. We are determined that the results of our collaboration reflect global diversity and therefore increase the possibility that we will solve serious mental illness problems that afflict all communities without regard to their status and location. So with that, I want to say thank you again, Dan, for inviting me. I hope this was of interest to the crowd. And I'm more than happy to answer questions now, or also if you want to email me. Thank you.
Thank you very much, Lori. One applause representing many.
I'll open the chat. I think I should probably open the chat. Yeah.
There are already a couple of questions on there from Anukrati's been asking throughout the presentation. So there's a bit on there for you to read. And anyone else, I'll just encourage at this point who has questions about any of Lori's research or her efforts, like tireless efforts to promote EDI or work on Ginger, anything else, go ahead, put it in the chat. Yeah, Lori, if you want to answer those questions, you just read them out first so that those who don't have the chat box kind of know what's going on.
Okay, sure. I stopped sharing because the chat's too small when you share, apparently. So I understand the studies have been historically biased. How likely is it that the patients from the minority populations are not giving their consent to the genomic data due to the risk of easy identification? That is an excellent point. I'm going to talk about this in 2 senses. One is domestic US. We're having this problem with COVID trials as well. There's a lot of distrust. Minority populations, there's a lot of distrust with the medical and research establishment. That distrust is absolutely warranted, considering the history. So a lot of this is, I would say, our biggest struggle getting people, not just consent, but to be part of studies is that initial barrier. In terms of easy identification, meaning like genomics data is basically your blueprint of who you are. You don't want to necessarily be identified. I haven't seen that as much in practice, I have to say. In our African settings, it's a slightly different than what we experience here in the US. And in the African settings, some of it is, as I mentioned, how do you explain what genetics is to a population that doesn't, where their local language doesn't have a specific word for genetics? So maybe you'll explain it as something like inherited coming from your parent. Well, that actually has very different connotations as well, and being able to overcome them. It's the answer to it is working within the community. I'm not the one that's consenting people to our studies. It is people within their population who are consenting people to their studies, people within their population that are talking with them about why what their contribution would be to this particular thing and what it's actually going on. So the bottom line is the answer to so many of my questions that I get is working with people within the community in order to actually best work with the community. I hope that satisfies a little bit. I appreciate the Ginger program. Thank you. Amazing initiative. Thank you again. I am wondering how translatable the program has been for the participants in international mobility for the temporary period in developed countries. It is often observed that many institutions in North America have a bias towards not acknowledging the quality of education received, if not in the US and Canada. Oh, this is great. I don't know who you are, but you have great questions. Should changes be made among North American university perspective for that matter to promote EDI? Yes. Okay, you asked me a yes, no question, so the answer is yes. They have to be made. There is a webinar that I have to look in my inbox for when it is. It might have already happened. I will send it to you, Dan, that is on this exact subject right now, having to do with journals. It's not just people in developed countries not respecting institutions, it's journals as well. So if my colleagues send in a paper and all of the affiliations say Uganda, Makerere University in Uganda, Addis Ababa University, it just doesn't even get reviewed, right? So there's a term apparently called white bull. So they will find a white person to stick their name on the paper just so the paper can get looked at if they submit it to a major journal. And by major, I mean a journal. And it's a huge issue of this idea that, well, it's coming from here, so it must not be of great quality. I also want to take a moment and address one other thing you said, which is acknowledging the quality of education received is not up to it. That is also another thing that I've encountered a lot of interesting questions that come to me, and sometimes people will ask, and sometimes people will ask, well, you know, you teach at Harvard and you teach there. I mean, like, come on, you know, like the students are different, right? And the response is no, they're not. Like I have taught at so many different institutions in the US and now in Africa and in Eastern Europe and a few other places. I have never seen the enthusiasm for learning as I do sometimes when I go to some of our classes in East Africa and South Africa. And Dan, you know, maybe you can comment on this as well, but like, it's not the lack of ability, it's not the lack of knowledge, it's not the lack of determination. Sometimes it's just holes in the foundation that you need to fix. And so, yeah, if they don't do it, if we don't start addressing these inequities, we're not going to be able to fix it. But to say that, well, they just don't get as good quality education, so like to not appreciate what they can contribute because they didn't have access to the same schools, et cetera, that we might have, is just leaving so much on the table that, you know, you can work with. Meaning some of my favorite collaborators, some of the most productive collaborators, some of the best insights I had are from collaborators with different backgrounds as mine. And I would say also that I can't go to Uganda and study schizophrenia. My psychiatrist friend, colleague, can't go to Uganda and study schizophrenia because he doesn't know what schizophrenia looks like in Uganda. If I want to be able to understand the disease in that population, the people that understand the disease in that population live in that population and that community. And so we need to engage them as well. And sorry, I just opened it, Dan. Maybe you want to comment and then just kept talking as usual. But you might have a comment on that as well in terms of like education inequity between our different places that we've been to.
I would only echo what you said, and you know, we've talked about this before. I mean, it's a very different experience teaching in Uganda than it is at Boston University during the summer, and you know, you do get there's a passion, you know, and there's a real urge for discovery you don't get sometimes when you're working within certain populations. I think privilege has a lot to do with it, background has a lot to do with it, and it becomes really apparent. Anakrati says, thank you for answering questions. That was great. They were great questions. We've only got 1 minute left, but I wanted to ask you something, not to put you on the spot, something maybe you could leave us with as a center, because you know, your work and everything that you've achieved is on such a grand scale, and you know, as you point out, takes years and years and years, and just, you know, a huge buy-in investment of individuals and institutions and time. For us as a relatively new center, KCNI was kind of just fully brought up, you know, the PIs only really came on at the beginning of 2019. So we've been alive and kind of grown really steadily for 2 years. And we, as I mentioned to you, kind of want to prioritize EDI and make a difference in our educational initiatives, our training initiatives, everything. Are there any lessons you've learned or any kind of key contributions or translatable elements or ideas that you might have? Again, not to put you on the spot, you know, for kind of developing our new EDI program, but, you know, we've had some discussions. I'm wondering if you had any kind of insights and thoughts on how we can start, you know, perhaps more locally or collaboratively in Toronto or across Canada or internationally, you know, but where do we start if we want to develop this as a group or as a center?
Oh, I mean, that's a wonderful question. And you're not putting me on the spot. I will say it's harder now, and it will get easier, hopefully, again, once COVID is a thing of the past. But you start small. So you think about putting together a new project and a new grant, and we're trained to think about research and what's the research question and what goes in the budget and all that sort of stuff. Think outside of that. Who's going to actually do the work? Who can I bring in? Is there money in there to make sure that I'm not just getting some postdoc that just graduated from this top level institution, but I can also bring in other people as well? I've been whispering in everybody's ear as they write grants, say, put some money in there for training, put some money in there for actual collaboration, like to build collaboration. So always keep it in the back of your mind. You can't always, not every mechanism allows people to do that, of course. But keep it in the back of the mind as you start doing it. Also, reach out and talk to people. International conferences now online kind of sucks. I'll say it. I like to travel, but it's also kind of good because you know how many of my African colleagues now are attending international conferences they never would have been able to attend before. So take advantage of that and start talking to people. I was going to send you someone, Dan, and he was actually in Toronto. There is a collaboration between University of Toronto and Addis Ababa University. He left when COVID started to go back. Benyam, you know Benyam, right? Doing a fellowship in clinical psychology in Canada, and that would have been a great. I was wanting you to maybe they can come talk and like talk about research and bring people in as well. So that's another thing. Like reach out to people, start conversations, continue conversations. That's how all of this starts. I also want to take 1 second. I see there's one comment from Jessica.
So Laura's asked a question. You can go ahead and you can.
It's not necessarily the question. She commented that she's a former Camp Glow speaker, and I didn't think this day can get any better. Like that was just a wonderful comment. Camp Glow was my first baby, and Moldova was the second country to do it, and that's where I was. And I just thank you so much. I'm happy to talk more. I know it's past time already. I don't know what the usual MO is for this, but if you reach out, I'm happy to talk more because you also have a really good question. How likely is it these advances will actually benefit the populations? Yeah, that's the river of lava right there that I don't feel like crossing. It sucks, it's bad, but I will say certainly with the COVID response, with the vaccines, because a lot of these vaccines are being tested in places like South Africa and other places, they're trying to write in the agreements that whatever comes out of this research, let's say vaccines, will be X amount available to these particular countries, et cetera, et cetera. Whether they've been successful remains to be seen, but some of it is they're getting more aggressive. African researchers are thankfully getting more aggressive and saying, listen, you want to do a clinical trial in my country, at my institute, that if the drug is successful, then we want access to that drug for this amount of money. Whether they're successful at it, it's still a work in progress. Yeah, but thank you all for your nice words as well.
Thank you very much, Lori. So great to hear from you, and thanks everyone for attending. And yeah, we'll talk soon. If anyone has any questions, you can reach out to Lori. You can find her on the Ginger website. We can give you any information, message in Slack. And yeah, Have a good day. Continue. I hope your day gets even better, Lori.
Yay, President Biden, Vice President Kamala. Yay. Okay. Bye.
Bye, everybody.